So now let's generate an action potential. Right? Let's generate an action potential. Action potentials only occur on the axon. Okay? So if on an exam, if I'm asking for an action potential, you're only talking about the axon. Okay? If I'm talking about a myelinated axon, right, you're talking about the magic that's going to be happening at each neurofibril node, okay, node of Ranvier. Okay? So when it comes to an action potential on a myelinated axon, we're only talking about this phenomena at each node of Ranvier. Okay? Because that's where my voltage gated channels are going to be exposed. Where the Schwann cell is, right, I'm going to have it wrapped in fat, wrapped in that myelin. Okay, so it's going to insulate those channels. So they're not going to be effective at stimulating and carrying that electricity across the surface of the axon. Right? If this was a unmyelinated axon, now we're going to be talking about all of these gated channels at every little stretch along the length of that axon. Okay. So that is something to just remember, even though I don't specifically have that down here. Okay. Action potentials only happen on an axon. If it's myelinated, at node of Ranvier to node of Ranvier. Okay. If it's unmyelinated, it happens along the entire stretch. I've reached threshold. Okay. I had a graded potential travel across the dendrite. I had a graded potential open up a sodium voltage gated channel. And it was enough to travel across the dendrite, travel across the nerve cell body, and get to that axon hillock. At that axon hillock, I have my greatest concentration of sodium voltage gated channels. I've opened them up. I got to threshold. And now this electrical current starts to travel down the length of the axon. Opening, triggering, if you will, voltage gated sodium channels to open. Sodium rushes in, flip-flop a charge. It travels across the surface of the axon, and it travels through the axon. Travels along the top of the axon and travels through the axon. We see it visualized on the top, right? So we talk about it, jumping from one node of Ranvier to the next, right? That's how it's, saltatory conduction. That's how we visualize it. But really, in reality, it's not just, hop, skipping, and jumping. It is traveling across the surface. That's where the damn channels are. But it's also traveling through the axon as well. Right? It's also traveling through that axon as well. That's why big axons that are myelinated transmit an action potential really fast, like 224 miles an hour, okay? Upwards of. They don't all go that way, but they can be really fast. Okay? So as that cell depolarizes, more sodium channels open. This is enough to be a positive feedback mechanism, right? To allow this membrane potential to completely become positive. If I reach threshold, that means stuff happens no matter what. If I reach threshold, it's an all or none type of response, right? The muscle contracts or it doesn't contract. An action potential occurs or an action potential does not occur. I've gotten a threshold. It's going to happen. Okay? This is a positive feedback mechanism. The electricity can travel along and through the axon enough to open the next voltage gated channel and the next voltage gated channel. Yada, yada, yada. Okay? Where I get to that plus 30 millivolts. where I'm getting electrical activity happening. I'm getting electricity to travel down the entire length of the axon. This is our depolarization. Okay, when I get to that plus 30. What happens when I get to plus 30? Again, sodium voltage gated channel inactivates, then closes. Potassium, good gosh, potassium voltage gated channels open up, and I repolarize. Okay. When sodium inactivates / closes, and potassium channels open up, this creates a negative feedback loop. Okay. Why is it negative feedback loop? Because the sodium channel right there opens up, and that causes an inhibitory effect that the impulse can't travel across anymore, right? And so that closes down that other sodium voltage gated channel, and potassium opens up. And this creates a negative feedback loop down the entire length of that axon, closing those sodium voltage gated channels. Inactivating, then closing sodium voltage gated channels. Okay. So an action potential only occurs on an axon. And if it is on an axon, then is it myelinated or unmyelinated? Okay. If it's myelinated, I'm only opening up those voltage gated channels at the node of Ranvier. For sodium or potassium. Okay. Magic is only happening there. If it's an unmyelinated axon, it's happening along the entire length of it. Right? Happen along the entire length of it. So there'll be more of those voltage gated channels having to be stimulated to allow this action potential to go. With that said, at threshold, there's been an impulse traveled across a graded potential that's traveled across the dendrite, traveled across the nerve cell body, stimulated all of those sodium voltage gated channels at the axon hillock. Minus 70, minus 55, plus 30, real dang quick, and boom, off and running down the axon. And it is going to travel all the way down the axon. If I reach the axon hillock and stimulate it, go from minus 70 to minus 55 to become stimulated, and then go all the way up to plus 30. If I've done that, the AP goes. The only way the action potential does not finish traveling down the axon is if you have one of those demyelination disorders. Okay, if you have MS, Guillain-Barré syndrome. Okay, that is the only way an action potential does not travel down the entire length. It is an all or none type of event. Travels across and through the axon. Able to travel through enough, and it's strong enough to stimulate the next sodium voltage gated channel at the next node of Ranvier. Continues down through. Stimulates the next sodium voltage gated channel at the node of Ranvier. As this thing is going down the length of the axon, back here at the beginning, sodium voltage gated channel at the axon hillock got to plus 30 and then inactivated and closed. And as soon as we got to plus 30 at that axon hillock, potassium voltage gated channels opened up. And this creates a negative feedback loop. Now the electricity stops propagating across and through the axon. And as we reestablish resting, the next sodium voltage gated channel there closes and activates / closes. Sodium voltage gated channel opens up. Potassium voltage gated channel opens up. Okay. And that creates establishing a resting, and electrical impulse stops traveling through and across. Closes that next sodium voltage gated channel at the next node of Ranvier. Potassium channel opens up. And we continue this down. It overshoots it a little bit. Yes, it hyperpolarizes, but there's sodium potassium pump to save the day and help to establish resting membrane potential. Okay. And that's all they're trying to say here. It's a positive feedback loop. Okay. When sodium voltage gated channels open and we get to threshold, sodium diffuses in and it keeps diffusing in and it stimulates more sodium voltage gated channels. Okay. So there's our stimulus. Okay. Right there. It was enough to reach threshold. Fully opening up that sodium voltage gated channel to the point where it goes all the way to ding dong plus 30. Again, sodium is the man involved here. As soon as I reach plus 30, that sodium voltage gated channel inactivates. When that inactivates at plus 30, potassium voltage gated channel automatically opens up. And it starts to rush on in. And here you can see it starts to drop pretty dramatically. Just as it did dramatically going up, it dramatically drops down. There's no sodium coming in anymore. Right? Potassium's rushing out, reestablishing the negative aspect on the inside. As potassium channels stay open and potassium comes across, at minus 70, they start to close. And again, there's my hyperpolarization right there. Just going to put HYP, hyper. Okay. I have hyperpolarized it. Don't like that. Okay. I'm closing up shop. I'm establishing resting. My sodium potassium pump kicks in. Okay. Minus 75, minus 80 millivolts kicks in. And it gets us back to our resting membrane potential. If I can't establish the RMP, the stimulus to get this nerve to fire again is going to have to be even greater. Does everybody see that? Instead of minus 70, let's just say it's minus 80. So it's going to need a greater stimulus, right? To get it to fire, electricity to travel down it. So I have to establish RMP with that sodium potassium pump, or the system doesn't function properly. And there you can see the picture down here gives you the milliseconds. You can see very quick to close completely for sodium. As you can see, potassium takes a much longer time to close, to open and close. So again, that's why we overshoot. It can get down to as much as minus 85 millivolts. If you want to remember that number, you can. Knowing what resting is, knowing what threshold is, knowing what the peak electricity is, that's for me most important. Hyperpolarization is anything more negative than minus 70 millivolts. Okay, but you can see it can get down to as much as minus 85. And again, like I've said 15 times now, it's a sodium potassium pump that does that helps to reestablish our resting membrane potential. This is an all or none event. Okay, an action potential is an all or none event. You don't have an action potential, get halfway down the axon and be like, you know what? I'm not feeling it. I'm going to stop. Okay, that never happens unless MS Guillain-Barré, some kind of demyelination disorder. Once that threshold has been reached, the threshold has been reached. Okay, The size of the stimulus does not affect the size of the action potential. Okay. It can be the minimum that slowly gets us there to minus 55 millivolts. You can increase the amperage and get it to go from minus 70 to plus 30 really damn quick. Okay. But an action potential is an action potential. That size of the stimulus does not affect the action potential. We always get to plus 30. Sodium voltage gated channel inactivates and then closes. Potassium voltage gated channel is always open. Is everybody with me there on that? At plus 30. Sodium voltage gated channels always inactivate, then close. At plus 30, potassium always opens. Voltage gated channels always open. Everybody with me there? Okay. So the size of that stimulus coming into that nerve doesn't matter when it comes to action potential. Okay. The action potential always lasts the same length. Okay. Always gets to plus 30 and deactivates. The size of that stimulus, the strength of that stimulus, all that means is that the frequency increases. So the stronger the stimulus, as you can see, all of these suckers are all the exact same height. Okay, this was a small stimulus, didn't do a damn thing. This is a little bit bigger stimulus. Okay, and you can see the duration here that it lasted. Here I generate an action potential. An action potential. Oh my gosh, look at that. All at nice prescribed, these are all equal. Here's the initialization of the stimulus. Okay, and I maintain that stimulus the entire time for this duration here. Okay. And even though I kept my finger on the trigger, the AP never rose above plus 30. So that action potential, it's always an action potential. Always goes to plus 30. Gets to plus 30 and then resets itself. Stimulus is still there. Well, then hell, I'm going to generate another one. Gets to plus 30 and resets itself. Stimulus happens again. Okay, because remember it, overshoots, comes back up, and then wha-bam! Overshoots, comes back up, and wha-bam! Keep that stimulus for an even longer time. You can see the longer the stimulus is there, the greater number of action potentials. Okay, and that's frequency. Okay. So the stimulus has nothing to do with the size of that action potential. The action potential is the action potential. Okay. But that stimulus can increase the frequency for which an action potential can occur. The bigger the stimulus, small, medium, large, the greater number of action potentials. Okay. Hell, in some instances, we can activate more and more neurons. Okay. This is referred to as recruitment. And you see a lot of this in a number of processes within the human body, okay, where we recruit more and more nerves. Memory is kind of an example of it. Okay. The more times you look at the material over and over and over again, you increase the frequency at which you are looking at it. You increase the stimulus of which you're going to activate neurons. Okay. That's why, they suggest, looking at something and saying it out loud and writing it 3 different times. That's a huge stimulus. You're using your eyes or using your ears because you're using your mouth. You hear yourself talking and then you write it down. That's a huge amount of stimulus in there to that region of the brain that's involved with memory. That's a huge stimulus. You generate a number of action potentials, and you help to strengthen the synaptic connections between those nerves. And that's what helps you create a very, very, very strong memory because I've increased the stimulus. Instead of just looking at it and reading it silently, now I'm going to say it out loud. Sodium voltage gated channel reaches threshold of minus 55, opens up more sodium voltage gated channels, gets to plus 30, continues the propagation of it. And now write it down as you're saying it and reading it at the same time. Okay? That's a huge stimulus, right? So this ability to increase the size of the stimulus doesn't necessarily increase a more potent AP, but it does create a greater number of APs. Okay? An action potential always stops at plus 30. Always stops at plus 30. When we do, we have to. When we get to plus 30, I've got to reset myself. Okay? Got to reset. Otherwise, I can't have another nerve impulse travel down the length of that axon. Okay? Refractory periods are after an action potential, and AP is action potential. APs are action potentials. Okay. So once we've reached that plus 30, again, we can't go any further. I have to reset myself. All right. This is the refractory period. All right. This is when I'm inactivating that sodium voltage gated channel and I'm opening up potassium voltage gated channels. All right. This is when a nerve cannot be excited again, this refractory period. All right. And refractory periods are necessary when it comes to resetting RMP. As you can see, I have a couple different types of refractory periods. Absolute and relative refractory periods. And this will be the same type of phenomena that occurs when, we're talking about muscles. This will be a different phenomenon when we talk about cardiac muscle. And it has to do with just two subtle differences that we see in cardiac muscle that we don't see in skeletal muscle. For nerves, we'll start here, absolute refractory period and relative. When it's absolute, it's absolutely cannot happen. Okay? Once a sodium voltage gated channel inactivates, can't pull the plug. Okay? It's going to go completely and reset. Going to go completely close, reset, and then unplug itself. So absolute refractory period occurs when those sodium channels are inactivated. Okay. A relative refractory period is dealing with potassium channels. Okay. Only potassium are open. Okay. The sodium voltage gated channel is completely reset. It's closed and it's unplugged itself. Okay, no plug. Plug's removed and it's reset itself. And it's just closed. Okay, so it's back to its normal resting state. But potassium is open and potassium's rushing out. Okay. It is during this refractory period where if the stimulus is significant enough, okay, I can get an action potential to start traveling down this sucker, even though potassium voltage gated channels are open. So one refractory period is due to one specific type of voltage gated channel, pretty much sodium, and it's the inactivated state. For relative refractory period, we're only involving one, that potassium. Even too, if you really want to split hairs with me and write it down and argue it out later, I'd take this argument as well. Absolute refractory period includes not only the inactivation of the sodium voltage gated channel, also the opening of potassium. Because at plus 30 millivolts, all that junk is happening. Sodium voltage gated channel inactivates, and then potassium voltage gated channels open. So if you wanted to say absolute refractory period is associated with both sodium channels, sodium voltage gated channels inactivating, and potassium channels opening, hell, I'd buy it. Okay, I'd buy it, I'd take it, I'd accept it. But that relative refractory period only involves potassium. Okay. Sodium is completely reset, and it can open if the stimulus is strong enough. And each time we send one down, it's a separate event. Each time we stimulate that axon hillock, we are stimulating this event to go. If I don't stimulate the AP at the axon hillock, nothing happens. Okay? An action potential is an all or none event. It is an all or none event that happens on an axon only. If I trip it at the axon hillock to go, it is going. If I do not stimulate enough sodium voltage gated channels to open up at the axon hillock, I do not establish an action potential. Again, action potentials do not travel down the entire width or even partial width and then stop. Okay, they go or they don't go. All right. So the kind of the last thing here is just the concept of what I've been talking about here. All right. This electricity is traveling not only along the surface of the axon, it's also traveling through, okay, as well. And that is something that you don't really think about. You just, use saltatory conduction, jump, 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 jump. And we just think of it traveling across the surface. But it does travel through the actual axon itself, the actual cable. And they refer to it as cable properties. Right? How electricity travels through a cable. It not only travels across the surface of it, but it travels through the fiber itself, through the cable itself. Right? And what do we have here? We have, protein fibers, right? We talked about anterograde and retrograde movement of stuff. So, there's organelles through here, helping to maintain the status quo of this structure. So there's things that are going to impede the movement of ions. And the smaller that is, the more times it's going to bump into stuff. All right? And that's going to slow it down.
So this is saying what exactly? That it occurs actually along the axon as well as the myelin around it?
Yep. It's going to travel, if it's myelinated, from node to node, right? And you see that saltatory conduction. It's that quote unquote jumping, right? That you see, that you think you see. But it also, it is traveling through the structure itself. Okay, so it's traveling through this axoplasma as well.
It's all doing it at the same time, right?
At the same dang time, you betcha. Okay. And obviously this is filled with cytoplasm, right? Cytoplasm, organelles, cytoplasm. I mean, it's watery component, but there's there's a whole bunch of stuff here, right? Can you run faster on dirt, or can you run faster if I put you in a pool?
Dirt.
Right. And if you can say you can run faster in a pool, then come on, let's go. Let's see it. I want you to videotape it. Okay. Maybe Tyreek Hill, but that's probably about it. So this is going to eventually, if it's not strong enough, you can see if I have a sodium voltage gated channel here and I have not one until right here. I mean, is this too far? Probably. Okay. Probably. So will this stimulate that voltage gated channel from opening up? No. But that's why we have sodium voltage gated channel a hell of a lot closer to each other in an axon. If we were talking about a dendrite, yeah, there is a huge amount of distance between them. Okay. We're talking about a nerve cell body. There is a large distance between them. Okay. So can that action potential dissipate? I'm sorry, graded potential dissipate before it stimulates the next voltage gated channel to open up and allow that to travel over a very small distance? Yes. All right. But in an axon, these sodium voltage gated channels, those Schwann cells are only so long. They're only so long for a reason. Because that's the amount of distance that it can just go, like the electricity can just go a little bit past. So this is my Schwann cell right here, let's say. Okay. That electricity can just travel just far enough to stimulate the next sodium voltage gated channel that's right there. And then there's my next little Schwann cell. And it's able to travel through enough right there to stimulate the next sodium voltage gated channel. Everybody with me here on this part?
Yes.
Yes, beautiful. It's clear as mud. At least as long as the words make sense today. Okay, as long as the words coming out of my mouth are making sense. That's really what I'm looking for. I know you don't fully understand this, and don't worry, I get it. You gotta look at this. Chew it, let it ruminate.
The only thing I'm still just a tad bit confused on is, so an action potential travels down the entire axon, or if it's myelinated, it just goes from node to node of around VA, or if it's unmyelinated, it happens still along the entire length.
Let
Am I
me
partially right for like?
let me show you the next slide. Okay, so you can get a better visualization here.